Thank you. Oh, sorry. Okay, so good afternoon, everybody. And so to reflect, I guess, the different expertise in the team, we've split this into three. And so I was going to start with a 15 or 20-minute overview, kind of what metagenomics is, thinking a bit about the microbiome and kind of the sort of bigger picture. Akis is then going to talk more specifically about the experimental approaches and sort of the lab-based challenges, and then Abraham finally will talk a bit about bioinformatics challenges and a little bit about some of the tools and more technical approaches that are available, and then there'll be some time for some questions at the end. And I guess some of this is going to be, I guess, not news to everybody, but I thought in the interest of really kind of stepping back a bit, it's interesting to think about what the microbiome is. And so just this really interesting idea that we're colonized by, or fact, not idea, that we're colonized by microbes. And there's lots of them. I mean, initial numbers were a bit out of kilter and put the proportion of microbes to human cells as 10 to 1, but more recent estimates put that about a 1, so a 1 to 1, 1.3 sort of to 1 ratio of microbes to human cells. So still, large numbers of microorganisms essentially inhabiting our bodies. And that leads to this last point, actually, about the characterizing the unit then as the wholly biont. And so we're not just individuals with our own genome, but we're actually, as a unit, very much, I guess, living with this interesting mass of cells, bacteria, and the viruses, and fungi, lots of things essentially living in our system. And then some would consider that a unit of sort of ecological or evolutionary unit, but that's maybe more controversial. And I guess as we kind of progress through the presentation, you'll see that there's really a lot of complex dynamics and interactions going on. Some people discriminate between microbiome and microbiota, but really those two terms have now started to be used synonymously. So really, there is some context to the way these different words are used. And I guess what's really clear from contemporary studies is this role of the microbiome in health and disease. And so disentangling causation and correlation is very important there. Metagenomics then is really the study of the genetic material in the microbiome. And so you're looking at metagenome sequencing to characterize all the microorganisms in a specific location. And that's usually done directly from a sample in an untargeted manner. And as Akis will allude to or demonstrate, there's a lot of technical hurdles there, and that's not as straightforward as one might hope. But really, I guess, the methods continue to develop. We're getting better at these kind of technologies. And the goal really then is this idea of an unbiased sample of all the genes and genomes from that microbiome, that microbial community. And there's some history to that. So on the left there in the slide, you can see that we sort of started with 16S ribosome RNA studies, and so that's very bacterial focused. To get into the virus, then we really do need to use shotgun metagenomics. We don't have the same marker gene sets, and so it's important to use metagenomics. There's also an area called metatranscriptomics. So actually looking at the active RNA in a sample, and people are starting to use these together. And even now we're starting to, there's studies really on the cutting edge that are doing single cell transcriptomics on microbiome samples. So really kind of looking individually into the bacteria, and this will be, I guess, very powerful for future virus studies where you're getting, I guess, direct information, not just about the community, but actually the intracellular information too, as you kind of move from a species co-occurrence ecological to a intracellular systemsy kind of understanding of these systems. And really, of course, what's happening then is we're generating these data sets, usually Illumina short read data. And so it's always important to, to disentangle then what is a biological system and all its complexity from the approaches we're using, the experiments that are giving us information about that system. And so those buckets of short read data then have to be put together using the computer bioinformatics to really try and, I guess, tell us information about that system. And I guess the thing to emphasize about bioinformatics is there's considerable challenges. There's a lot of real technical problems there. And the one, I guess, one of the main ones is just that we're highly dependent on reference genomes. So in order to characterize a new piece of genetic material, we're comparing it to something that's existing with viruses. They can be highly divergent, and we have a lot of uncharacterized data. And so there's real challenges in kind of making sense of the signals or the information we get from these samples. And it's really important to emphasize, and I guess us as a community are very aware of this, but the virome has been very undersampled in relative to the microbiome studies. And when you pick up journal articles and they talk about the microbiome, most of those are talking about the bacteriome, and so they're not really including the virus. So most of the published literature, as you can see here in that chart, is ignoring the virome. And so I guess studies like CURE are kind of I guess start of this newer focus on the complete microbiome, as it were. And so there's, I guess, interesting questions. So the first, just what's present in the sample, what bacteria, archaea, viruses are present in the sample. You may have fungi, you may have other, microorganisms, protozoa, etc., but we're tending not to include those in the analysis. A major question is what species are co-occurring. How do these relationships change through time? And just as that figure there that I took from a recent article emphasizes, what we mean by time here is actually complicated. You're talking about a system that's co-evolved with an individual from birth to old age. In our samples with asthma, we're usually sampling over weeks to months to years. And so there's many technical, I guess, challenges in studying the microbiome, not just that in a daily cycle, so in a single day, the system is changing depending on your diet, and depending on if you're outside, if you're breathing cold versus warm air. So very much it's a complex system. And there's also lags in the system. Things are changing slowly through time. And so we're studying, I guess, things changing both on an ecological community level where things are interacting with each other, particularly for viruses. They're targeting different bacteria. The eukaryotic viruses are targeting different host cells. And then you have on the other side of that an evolutionary axis. So very much the viruses are changing. They're changing the bacteria they can infect. Some viruses, eukaryotic viruses, as they use the cells in the system, are having to adapt to use different types of cells as they kind of evolve in the course of the infection. And so again, this is giving us this very complex system. You have time, evolutionary time, ecology, all coming together really in a complex system. And so when it comes to the actual analysis, then, so the computational analysis, the bottom line is it's complicated, and this isn't a problem that's specific to metagenomics, and that there's really just not a black box. There's not one way to do analysis, one right approach, and in the literature there's much disagreement. And I guess these are we shouldn't be pessimistic about these problems. These are signs of a healthy system. Bioinformatics, computational biology, is a relatively new discipline, only a few decades old. And it's very much a research field. And what's exciting about that is there are always tools being developed, new methodologies that are coming online all the time. What's frustrating about that is there's always tools, new methodologies coming online all the time. And so it can be challenging to stay abreast of what's a real innovation to something that's a more, I guess, incremental innovation. But broadly, if we look at that image there, you can see there are kind of, I guess, different categories of types of analysis. So from marker genes, very much specifically focusing on specific genes, to assembly approaches, which really just working with the raw data, doing things de novo, like trying to pull signals out of the data. And that's something that's really, I guess, in a lot of the recent high-profile microbiome studies, have really leveraged that de novo assembly approaches to pull out contigs, so long stretches of genetic material that are starting to get full viral genomes out of the data sets. And we're really, I guess, making big strides in that data. And the scale of the data that's been analyzed is leading to a lot of new information about new viruses, new bacteria. And so going forward, the field is very healthy. Other approaches that are very popular are binning, so using things like what we call alignment-free signals, just genome composition signals to, first of all, characterize data into bins that then allow you to apply the bioinformatics methods to those. And I think, as Abraham will illustrate with some of the tools that are available, it's a very vibrant area of research. There's no one way. And so very much best practice is taking a census of current methodologies. And that seems like a fairly, that's starting to settle down a bit as a discipline. And so there's many challenges. So you're obtaining context, not necessarily full genomes. Some of the species are at a low abundance, so it can be difficult to detect. If you detect something very low frequency, it can be an artifact. And I guess a particular challenge of viruses is high diversity. So what we're using when we do any kind of taxonomic classification is the notion of homology to say that this virus gene is evolutionarily related to another virus gene. The problem there can be we're not necessarily capturing a signal in the data for that. And so if evolutionary distance or evolutionary rates are very high, as is the case with viruses, then the sequence similarity signal we use to infer homology can be lost. And so we always have to be careful when we talk about homology, evolutionary relatedness. That's a binary signal. Two things are or are not evolutionary related. There's no degree to that. There's no amount of evolutionary relatedness. There is a degree of sequence similarity. And so that's very much the signal we're using to infer the other. And so with viruses, people are trying to think about using amino acids signals, so getting away from nucleotides where you may have saturated signals, and going above that, starting to use protein domains, even going to sort of secondary structures. And so trying to leverage into much deeper evolutionary distances to sort of properly classify things. As an aside, there is actually some kickback from members of the sort of taxonomic or the viral community who really, why do we need to name everything? And so I think that's a good point. We can detect things, particularly in longitudinal studies, as evolutionarily related and actually as taxonomic units that can be important or not. And whether that has a name or not, is maybe less important than to actually understanding the dynamics of the system. Another important point is really, I guess, pangenomes. So bacteria really aren't one genome. They're very dynamic systems, and the gene content is changing all the time. This is also the case with phage. Gene content changes all the time. So we do have to be careful that we don't overemphasize one particular gene set as belonging to a particular virus or host. Viruses in particular can recombine. Phage are particularly good at carrying genes from one bacteria to another. We have different lytic versus lysogenic states. And also rates of evolution can be influenced by things like having CRISPR-Cas systems. And so if the bacteria has a CRISPR-Cas system, that can influence whether its rate of evolution. And then finally, dark matter, just that uncharacterized space. There's a lot of things there that we just don't have names for. And so with longitudinal samples, we can really start to look to see, do we detect unknown things that seem to be present in multiple samples? And I guess this really brings us on to what I guess is important when we think about a microbiome and what it is and what its meaning is through time. And so in the microbiome from the gut, for many years there was a lot of emphasis on the core microbiome. And so comparing lots of samples to look for the sort of this idea of a stable core, and there's this name enterotypes given to this. But sometimes that's kind of an illusion, because when you focus on a sort of a static core, you're not picking up really what's changing through time. And this is a dynamic system. The bacteria, some of those are cooperating with each other. Some of them are interchangeable in the system in terms of sharing metabolites. The viruses are predating on the bacteria. Some of them have more commensal relationships or helping the bacteria. in terms of, I guess, how they respond to other viruses. And so really the system is in its normal state inherently dynamic. And Tucker recently found a paper where they called this a dynamic core, because we'd been sort of using the term dynamic normal, which is a horrible term. And so this idea of this dynamic core is very interesting, and that feels really important for our study, is having some handling on what the normal oscillations are of the system, what are the boundaries for that. Because once you understand those, you can start to get to grips with what a shift or a change really is. When is the normal steady state, the normal dynamics of the system altered, and so that you're maybe entering into some kind of potential disease state. And so Joe, who's involved in the study, is taking particular focus on the PhD student here in Glasgow on the interspecies interactions, and this will also feed into the mathematical aspects of the project with Tucker and Mark. And so how do you essentially determine the correlations, the interactions between the species in the system? And so at the simplest, if abundance of two species is correlated, assuming a positive relationship, if it's negative, a negative correlation, some negative relationship, and that fits into quite a nice classical ecological framework of commensalism, mutualism, and so how do things influence each other? And that's a very nice paper here that I've got these images from that discusses this, by Faust, who's done a lot of work in this area. And so again, as with the bioinformatics, when you get into this aspect of the project, studying the microbiome, there's a very healthy field and body of literature that we can borrow from. And the image on the right, it's not very clear, but it just shows the methods matter. Applying different correlation methods give you different results. And so again, it's really about, I guess, reaching some consensus of when change is significant and what change really means in some biological and sense. And again, then, so when we're thinking about change, there's a quite interesting model called the Anna Karina principle that I think Akis has talked about this quite a lot in his presentations, and that's the sort of, I guess, this idea of there being a resilient system that has a dynamic core that is resilient, and so that system can be pushed to certain bounds and come back to some normal. And so that gives us in our individual microbiomes some inherent stability. But then in a disease state, what's happening is the system is now going off in some fairly stochastic manner. And that's not particularly deterministic. That's now kind of unpredictable. It's difficult. You could look at people that maybe have a similar phenotypes in the disease states, but the microbiomes might not necessarily correlate. And so that's whether the kind of reality of this is or how close reality is to this is less important than it's just a model to work with, a way of simplifying our understanding of the system to generate a model. And so really, I guess what's important is really to appreciate that when we're studying the microbiome, it's a complex system. Metagenomic sequencing is the tool for getting together all of the relationships to that. But it's a complex system. There's a lot of stochastic processes going on in there. There's a lot of just random stuff happening. And so there's literature where, for example, people have shown that it's possible to end up in a disease state just through stochastic kind of chance outcomes. And so I suppose when we're really thinking about dysbiosis and disease, we're looking for kind of deterministic causative predictors, things that can be the understandable features that allow us to really get to grips in some way with what's going on and disentangling causation from correlation. And so we know many of these things. So pathogenic invasion in the context of things like asthma, rhinoviruses, damaged epithelial layers, antibiotic treatments. And so there are very understandable direct perturbations to the system that will influence the microbiome. And then our individual genomes, interactions with the immune system as we age. And so there's a complex set of interactions going on that really, I guess, relate to lifestyle, and that feels like we are kind of coming together now as a community to think about these systems, and in some ways this was very nice about this project. We have an eclectic set of individuals with complementary expertise to think about this complex system. And so Akis now is going to talk a bit really specifically about the bioinformatics challenges and the experimental challenges. Thank you.
Okay. Thanks, David. Nice presentation. So I will give a broad overview of most of the experimental bit of metagenomics and how we can tweak or customize these experimental pipelines in order to answer specific questions. So in this slide, we can see how we split the metagenomic pipeline in the wet lab and the dry lab. So broadly, metagenomics are split into two groups. We have shotgun metagenomics and target metagenomics. The aim of shotgun metagenomics is to describe a microbial community. This means that we don't care about looking at specific components like the bacteria or the archaea or the viruses. Rather, we aim into discovering whatever genetic information is present on the environmental side that we decide to sample. This means that we don't use a PCR based in any marker genes. And for most instances, a step where we need to amplify our genetic material needs to be included in the pipeline. In target metagenomics, on the other hand, we want to study a certain taxonomic group of microbes, for example, bacteria. In that case, we amplify our DNA libraries using a marker gene that we can find across all the species or the families of microbes that we target. We can also perform a whole genome amplification step coupled with PCR, and we can use target metagenomics when we are interested in a single species. For example, in cases where we have a viral infection, probably from a known agent that we want to identify the genome or the species that causes the infection. Use the okay. So in order to optimize the pipeline, there is a lot of different choices we need to make. And what is really important to understand is that even though we implement various quality control steps during the pipeline, since we don't know what's in there, what's on the sample that we sequence, we need to optimize everything. We need to start the experiment, wait for the sequencing data, analyze the data, and then give a feedback back on the experimental pipeline in order to optimize it. So this makes it extremely time-consuming and costly process. So these are over here is a list of, let's say, some major decisions or challenges we have during the experimental bit of metagenomics. And the questions start with the SOP, the standard operating procedure, the type of the tool that we're going to use for getting the specimen. Of course, this depends on the accessibility to the sampling site. We need to take steps in order to preserve nucleic acids and, of course, protect our sample from contamination with environmental microbes. Second important thing is the biomass. So we're talking about the actual biomass of the sample. This will define what type of nucleic acid isolation we will decide to do. It is affected by the contamination with host cells or with host genome fragments. And an important bit is whether we can actually define the sampling site in terms of area or weight, and this could give us a more absolute quantification of our metagenomes. The enrichment protocol, it depends on whether we're doing shotgun metagenomics or target metagenomics, as I told you before. In some 16S-based sequencing approaches, they tend to use some marker species for spiking samples. In the setting of cure, this is not something realistic because we have so many different types of viruses, and over here you can see just a small list of the different types of viruses we identified. So spiking a sample with a known viral sequence, it's in order to test for basically for bias, just something possible. In the isolation techniques, of course, we can do DNA and or RNA extraction or both. DNA treatment, when we look at the RNA portion of sequences. Of course, we can remove somehow human sequences. That's something we could do. And ribosomal depletion is also another option. However, for community-based metagenomics, if we deplete the ribosome, we will probably lose a lot of information coming from the bacterial component in terms of classifying bacterial genomes. But an added information of including actually the ribosomal subunit of bacteria is that we can distinguish between active bacteria and resting or dead bacteria. So as you can see over here, active bacteria have a much higher number of ribosomes per bacterium. So it's about 7,000 ribosomes, comparable to only 7,000. So there are approaches that try to use this type of information to discriminate between dead and active bacteria. And of course, all the above can affect the classification process of our sequences. So the yield of our DNA and RNA isolation is also important. This also depends on what type of analysis we want to do. When we're analyzing RNA, we need to take special care to use high fidelity reverse transcription polymerases to change RNA into cDNA, and then from cDNA to transform it into double-strand DNA in order to use it for DNA library construction and further sequencing. The whole genome amplification protocol is affected by whether we're going to do shotgun or targeted metagenomics or a combination of those in some cases of viral discovery. The type of the whole genome amplification polymerases is extremely important because we need to take into account the coverage of the targeted genomes and the stability of the DNA during the amplification process. The library preparation protocol depends on the sequencing technology and the available DNA material. So basically the quantity of DNA. For example, nanopore sequencing demands a higher DNA input than Illumina-based sequencing. Then the depth of our sequencing is also really important. So if we know, this basically it's back on the experimental protocol. So when we are able to define a certain sequencing depth, then it can define the type of sequencing platform we can use, what multiplexing strategy we're going to do, so how many samples per sequencing run, and of course all these define the cost per library. So when we started doing metagenomics over here, we started with a cost of it was about £500 for a metagenomic sample, doing both DNA and RNA sequencing. And now, with all this information we have gathered throughout the years, we have dropped this cost, for example, into about £320 per sample. So things are getting better and getting cheaper. But as David has shown you, the value for the cost for when you compare it with the amount of information we get is really comparable to 16S-based sequencing or other marker gene-based metagenomic approaches. For the bioinformatics analysis, Avram will discuss about these issues. Briefly, we do a quality check We clean up our libraries from contaminations, and then we do a direct classification or de novo assemblies. And the final step is we get a list of all the metagenome assembled genomes or our metagenomic libraries. So as I told you before, all the different bits within this pipeline can affect the final stage. So starting from the SOP, what we need to take into account, and especially for the respiratory tract, is that we know already from 16-base sequencing that different parts of the respiratory tract, they have different environmental niches. So some factors that define these microbiome niches is the existence of mucus or ciliated cells, oxygen supply, blood supply, the temperature, the existence of metabolites that can act as a substrate for the different bacteria, and of course the pH of the site. This we need to take into account when we try to access the specific environmental site in order to avoid passing through different environmental niches. We also need to take into account the accessibility for environmental microbes to reach the specific environmental site that we target. And in Cure, we already developed some computational pipelines where we can discriminate between environmental microbes and the microbes that are actually resident in the targeted area. Of course, we need to take into account the let's say ketome, all the buffers, all the kits that we use. They have some low level of contamination. With, of course, a very limited diversity of different types of species. We use negative controls, which follow the whole process from the point we get the swab up to the point where we sequence our libraries. So we can account for these contaminations and remove them. As I told you before, if we can define the measurable area, that's a plus, because we're closer to getting a true realistic snapshot of the metagenomic community. And we're working now into some ways where we can use the host sequencing reads that we get as a contamination in our samples in order to find sequences that are shared between samples, and then design a mechanical assay that will manage to mechanically remove all human sequences. And I will come back to this. Preserving the specimen, so nucleic acid integrity, and if we care about preserving infectivity of some viruses, again, there are specific protocols for this. Avoiding free storage and is really important. We need to avoid degradation of DNA and RNA, and reassure that we have stable nucleic acids. And the final bit is it's important whether, or is a question that we usually get, whether there's difference between staff collected versus self-collected swabs. And there's been a couple of papers that they have tested this and they didn't find any real differences between the microbial communities. So I think in Cure, where we have some self-collections, now we have reference papers that we can use for this. Now, host contamination. Host contamination is the biggest, I would say, problem in respiratory metagenomics. Over here in this graph, you can see different tissues, and you can see the level of human contaminating sequencing reads. And as you can see over here, the nasal cavity, for example, it has more than 90% of human contamination. So when we compare it, for example, with stool samples, where basically we can just take the DNA and RNA and sequencing directly without caring really about if we have any contamination from human cells or human sequences. And this pie chart, this comes from actual data from our pilot studies. So this is a picture before we do any type of processing. So that's the actual contamination we get from human sequences, and then we have just a little bit like about less than 5% of microbial species. Why is this important? So human genome is roughly 1,000 times larger than an average bacterial genome. So even though we might have more bacterial cells, this is not the same for the humans, for the genome sequences. and take into account that most nucleic acids that we identify in the respiratory tract are naked, which means they're not basically protected by anything, or they're not within a cell. Then this creates this huge problem. So the percentage of sequence reads aligned to the human genome varies widely by sampling site, as we show in the graph. So now we're moving into the different protocols there are out there that have been published up to now, different ways we can enrich our libraries, our metagenomic libraries for microbes. Each one of those has given fruitful results in different applications. Generally talking, whenever we see ultracentrifugation, this refers usually when we want to enrich mostly for the viral component of the metagenome, but this is extremely biased towards DNA viruses. So it's not something that we can make use of when we want to have just the community analysis of the microbiome. Detergent treatment, again, it introduces some biases towards certain types of viruses or types of microbes. Low-level centrifugation, that's something that, as you can see, it's common. It's really important to bring down cell debris and cells, intact cells, so we can use the supernatants to proceed with the isolation. Filtration, there are some papers they have shown some better results using a filtration system, but others they show that there's no difference. Important thing is nucleic acid treatment. So mostly it's a DNA treatment. If in cases where we are interested into isolating intact virions, this can be used. So basically we destroy all naked DNA and we retain just whatever has been encapsulated on a virion. But as we have seen in our preliminary data, most of the nucleic acids in the respiratory tract are naked. So by using a DNA treatment step, we bias our metagenomic libraries. Gradient centrifugation it's ideal for target metagenomics, especially when we know the infectious pathogen. However, it is affected by the physical properties of the virions. As I told you, it's biased towards double-strand DNA viruses. The segments are not easily defined, so basically this will increase the number of samples we need to sequence. And we can have cross-contamination between the segments, so something that is totally targeted. Whole genome amplification is a very important step. There are different types of whole genome amplification reactions. Multiple displacement amplification is the one that is generally preferred, and that's something we use. And we can have multiple displacement amplification without any specific PCR primers or with a combination of those. So a few words now about the polymerase that we use and their effect on the metagenomic process. So it's really important that we have a very good amplification of even very small amounts of DNA. So over here you can see the input number, amount of DNA, and with blue you can see the amplified amount of DNA. And as you can see here on the scale, we're talking about minute amounts of DNA input and amplified DNA. This is really important. This gives us great analysis, and we use this type of polymerase within our data. Also not showing any particular biases depending on the type of the DNA molecules. Coverage, efficient coverage of the target sequence is really important. So over here is the sequence, target sequence, and over here and you can see the coverage of the sequencing reads, whereas over here with different colors we have different polymerases. So you can see the one that imports, let's say, the less bias when you compare between different polymerases. This is important to get a good identification of our genome fragments. Over here again you can see the effect of the polymerase on genome coverage. So this is the sequence genome, and this is the same. You can see the accumulated fraction of reads against the accumulated fraction of genome, and this is the distance when we use different polymerases for amplification process. Over here, you can see the effect of whole genome amplification polymerase on coverage uniformity. Again, this is our target sequence, and then you can see the use of different whole genome amplification polymerases, how they affect the coverage uniformly. Finally, we can see the effect of this polymerase on the GC content. Here, this is the GC content of our target. And by using different types of whole genome amplification polymerases or different amounts of input DNA on the whole genome amplification reaction, we can have a bias towards the neutrality region, and this will limit our power of detecting or classifying sequencing reads into microbes. So choosing a sequencing platform is really important. We use Illumina. As you can see in this list, we have different platforms. Each one of those, they have, the pros and the cons. So Abraham will talk more about this. A really important bit is defining the appropriate sequencing depth and length. So we need to take into account, let's say, this list of 7 major points. First is representation of all 3 superkingdoms and viruses. The representation of all genome types, whether we want to do diversity and taxonomic variability tests to check for the genome content, do functional annotation of the microbial community, look at ecological patterns or evolutionary patterns within our metagenomes. Depending on what kind of questions we want to answer, this can affect the depth and the length of our sequencing. So as I told you, we have used Illumina. We have used essentially most of these platforms: Miniseq, MiSeq, NextSeq, and HiSeq. Currently, we're using HiSeq, so we have very good experience with different platforms, and we also want to test with Nanopore in future steps. So the effect of sequencing depth in discovering viruses. Over here, you can see different sequencing depth and sequencing read lengths. When we compare MiSeq versus HiSeq, we're looking at the virome, and you can see the effect of the different platforms on the eukaryotic and the prokaryotic viruses, both on their occurrence, so on our ability to detect them, but also on the diversity. And basically, what we're doing when we try to adjust our sequencing depth is basically it's like fishing. We have different types of fishes in the different sea levels. So with our processing, we drop our host contamination on average of about 60 to 70%. So this allows us, by using about 15 gigabase pairs per swab, to have a very good description of bacteria, fungal, viral, and archaea species within our metagenomes. Over here, you can see for the DNA and the RNA, you can see the different samples, the output number of reads, the reads that survive our quality checks, tests, and over here you can see for each sample, so for each swab, the DNA and the RNA derived microbial sequencing reads. So we have lots of information. And that's the final, our aim is basically with the same way that we do denaturing gradient electrophoresis, for example, we use 16S-based marker genes. We create this virtual metagenomic profile where we don't only have bacteria, but we have all different components of the microbiome, and then we can use this information to answer all the different questions. And I will not go into any data we have or results, and just to show you this, when you relate this with picture I saw with fishing, over here you can see the relative abundance of bacteria, eukaryote, archaea, and viruses on the same sample. So this is a snapshot of an nasopharyngeal sample and its composition into the different major components of the microbiome. So that's it from me.
So yeah, and lastly, you have a number of tools that allow you to basically use the information from the assembly process or the alignment process to or the taxonomic classification process to build trees, compare different metagenomic samples together, identify similar profiles, or even cluster your sequences or metagenomes based on their behavior over time. Now, there is no one solution fit all cases. So as David mentioned, you have to go based on best practices and what you want to achieve, and build your own pipeline based on that. And our pipeline for actually trying to identify all the species, or as many species as possible that exist within the as microbiome, is as follows. We have the data that we clean using Automatic, for example, and then we move the human host, the host reads from our data set. And in this case, because we know that our host is human and we are not interested with human, we use the human genome as provided by the NCBI database and bow tie aligner, and we're aligning all the reads to the human genome. The ones that actually align to the reference are being discarded, and the rest of the remaining reads are being used are considered as our respiratory metagenome. The next step, we use taxonomic classification tools and the NCBI database of reference genomes to split our remaining reads into 5 groups, or basically this is a supervised binning approach. And the reason that we're doing this is we split all the data into these bins just to make the de novo assembly process easier. And the groups that we are splitting our data to are the archaea, bacteria reads, eukaryota, viruses, and the reads that actually cannot be classified as anything, or can be considered as dark matter, or can be considered as, erroneous reads. We don't know at this moment, so it's just putting them in that category. Then we assemble each group, the reads in each group, and then we extract the context from the assembly process, and then we align our reads to the de novo context that we obtain in order to eliminate any junk assemblies, assembled context. The final step is to again run taxonomic analysis on the de novo context that we obtained, and we consider that as not junk and try to put a taxa ID on them, and then work with this information further. From initial the use of our pipeline on the Cure longitudinal samples, we've managed to extract context with a cumulative sum of more than 400 million bases long. So we are getting something. We're getting a lot of information out of our samples. We use this information to identify relations and correlations between different species, how some species change in terms of abundance over time. Identify species that are, consistent in all of our subjects samples. Try to build a personalized microbiome profile for each patient. We also try to see if we can spot any changes in how asthma is progressing over time, and also, try to identify all the shared species among oral samples. Basically, try to see, can we identify species that are prevailing in all patients the whole time, and just try and see if there's something that drives that towards disease. So basically, that's the bioinformatics side and some of the problems that are facing and some of the solutions and tools that we can use to combat these problems.
Okay. Let's see now. We don't need the screen anymore. I don't know how you can take it back, Christo. Okay. And I mean, I understood very well that this is complex. That's the main message. So this is open for discussion.